Life cycle of retrovirus. Group-specific antigen, that is GAG proteins, are the genetic material that codes for the core structural proteins of a retrovirus and was a major component of the viral capsid. They are about 2000 to 4000 copies per virion. The pol gene encodes a 3 enzyme, protease, reverse transcriptase and integrase, that catalyze the steps of retroviral infection. The first step of replication is the binding of the glycoprotein to the receptor protein. The cell membrane degrades when they bound it and becomes part of the host cell, and the RNA strands and enzymes go into the cell. The core disassembles partially, allowing the virion copy of reverse transcriptase to start using the RNA genome as a template, and the conversion of retroviral RNA to proviral DNA, and is catalyzed by reverse transcriptase, and is necessary for proviral DNA insertion into host DNA, a step initiated by the integrase enzyme. Reverse transcriptase also has an RNase H activity. Reverse transcriptase also has an RNase H activity by which it digests the RNA genome strand. Thus, a transformation of RNA into DNA takes place in 2 steps. First, reverse transcriptase enzyme copies the RNA to form a RNA-DNA hybrid. Then the ribonuclease H component of reverse transcriptase degrades the RNA strand to leave DNA alone. After synthesizing DNA, the reverse transcriptase copies this strand to produce a double-stranded DNA called proviral DNA, which can direct the synthesis of mRNA and new RNA virion genome copies. Transfer RNA is carried by the virus and serves as a primer required for nucleic acid synthesis and facilitates immediate conversion of viral RNA to double-stranded DNA by the action of reverse transcriptase. Hence, the net result is a double-stranded DNA molecule that is actually longer than the RNA genome. Both ends of the DNA contain LTR, that is long terminal repeat sequence, whereas the RNA genome had some of the sequence at its 5' end and part at its 3' end. While this DNA synthesis was proceeding, it is likely that the overall complex was being transported towards the nucleus. Regardless of exactly where the double-stranded DNA is when it gets completed, it indeed is then transported into the nucleus, still in a complex with a part of the material from the original viral code, in particular the enzyme integrase. The actual mechanism of transport into the nucleus is known to be different in different retroviruses through a nuclear pore for some, such as HIV, while for others nuclear entry requires the breakdown of the nuclear envelope associated with cell division. Once converted to double-stranded DNA, the DNA enters the nucleus and is integrated into the host genome. The integration is catalyzed by a virally encoded integrase. Integrase becomes bound to both of the LTRs, thus holding the double-stranded DNA in a circle, and also interacts with a site in the cellular chromosomal DNA. The enzymatic activity of integrase then physically incorporates the viral DNA into the chromosomal site. Early experiments had suggested that the selection of chromosomal site for integration was random. Integration of viral DNA into host DNA is mechanistically similar to the insertion of transposon in bacterial chromosomes. For example, a few base pairs of host DNA become duplicated at the site of integration, forming short repeats of 4 to 6 base pairs at each end of the inserted retroviral DNA. These inserts are transcribed by the host enzyme into fresh RNA molecules which re-enter the cytosol, while some are translated by host ribosomes, while other RNA molecules become incorporated into fresh virus particles. Please see the figure on screen. The retroviral genome also contains a packaging signal sequence P, which is needed for the newly synthesized RNA molecules to be incorporated in fresh virus particles. Please see the figure on screen when a retrovirus infects a cell. Retroviruses have also provided essential tools for biotechnology. Reverse transcriptases from avian and murine retroviruses are used universally to generate cDNA copies of RNA, which then can be manipulated with relative ease for cloning and sequencing. Modified retroviral genomes are widely used in transient and stable expression of cloned genes in vertebrate cells. Because Reverse transcription lacks the usual proofreading of DNA replication. A retrovirus mutates very often. This enables the virus to grow resistant to antiviral pharmaceuticals quickly and impedes the development of effective vaccines and inhibitors for the retrovirus. Nowadays, scientists use retrovirus as vectors in gene therapies. If it successfully happened, retroviruses can be engineered to carry genes into the body. When retroviruses have integrated their own genome into the germline, their genome is passed on to a following generation. These endogenous retroviruses, ERVs, contrasted with exogenous ones, now make up 5 to 8% of the human genome. Most insertions have no non-function and are often referred to as junk DNA. However, many endogenous retroviruses play important roles in host biology, such as control of gene transcription, cell fusion during plasmid development, in the course of germination of an embryo, and resistance to exogenous retroviral infection. Endogenous retroviruses have also received special attention in the research of immunology-related pathologies, such as autoimmune diseases like multiple sclerosis, although endogenous retroviruses have not yet been proven to play a causal role in this class of disease.